Hi friends, today we are going to see the past year NEET questions for the chapter Biomolecules. It's a continuation here. Today in this video we will cover part 4. Now let's begin with the first question here. This question was asked in the year 1988. In double helix of DNA, the two DNA strands are. So this is an easy question for you to answer. So when we say double helix, of course, it has two strands, right? That is what is the meaning. In a double helix of DNA, the two strands are dash. How are they present? Either they are coiled around a common axis, or coiled differently, or around each other, or over a protein sheath. So here if you see, when we talk about DNA, the full form of DNA is deoxyribonucleic acid, and DNA is the predominant nucleic acid which is present in prokaryotes and eukaryotes. It's a long molecule that will contain a unique genetic code, or you can call it as a, sometimes they denote DNA as a recipe book, because it is the one which creates a new generation. What do we do with a recipe book? We create dishes. So that is what it does here as well. So apart from that, DNA is short. DNA in short, if you want to explain, it's a part of organism and it has coding and non-coding sequences. They do not code for protein sequences. Some non-coding DNA is transcribed into non-coding RNA molecules such as transfer RNA, ribosomal RNA and regulatory RNA. Now, in DNA double helix, the two DNA strands will be coiled around a common axis. Okay? It will not be coiled differently and all. The two chains comprising the DNA are spirally coiled around a common axis to form a regular right-handed double helix. Okay, so if you see, it follows the right-hand double helix. Can you see the image to your right side? Right? It has a nitrogenous base, a sugar, and a phosphate group. So this is what is here. Therefore, when it only when it coils around a common axis, you'll get the right-hand principle there. Therefore, the correct answer is option A. Next question here. This question again asked in the year 1988. RNA do not possess this dash. A very easy question for you. As I already explained, RNA and DNA are actually differentiated with the help of the bases. Right. When we talk about RNA, we have uracil. It will not have thymine. Therefore, the correct answer to this question is option B, thymine. Okay. So RNA has 4 bases. Adenine, so the diagram clearly tells you here. So you have adenine, guanine, cytosine and uracil. Instead of thymine, it will have uracil in its place. Easy question, right? Next question here, which was asked in the year 2017. Which one of the following statement is correct with reference to enzymes? So what is enzyme? What is the other name for enzyme? How can we define enzymes? See, enzymes are also called as biocatalyst, right? Enzymes are biological molecules. Usually they'll be proteins, okay, that is significantly used to speed up the rate of chemical reactions that take place within a cells. So this can be in coincidence, or you can compare it with catalysts that we use in chemistry for reactions, right? They will not interfere with any reaction, but at the same time they will see to that the reaction is speeded up. Okay, it enhances speed. Then it is also called as biocatalyst. Okay, so enzymes are biological molecules and mostly they will comprise of proteins. Okay, they are vital for life and serve as a wide range of important functions in the body, especially for aiding digestion and metabolism. So their main role is in that. Now coming back to the question here, I just gave you an introduction of what is enzyme is, because you should know, right? Now, which statement is correct here? And look at the first statement here. Holoenzyme is equal to apoenzyme plus coenzyme. Coenzyme is equal to apoenzyme. See, option B is wrong. Coenzyme cannot be a composition of apo and halo. Halo cannot be a composition of coenzyme and cofactor. Wrong. D is again wrong. Therefore, the correct answer is option A. Haloenzyme is conjugated enzyme which will consist of a protein part called as apoenzyme and a non-protein called cofactor. So, coenzyme. Coenzyme is also an organic compound, but their association with apoenzyme is only transient and acts as cofactor. What do you mean by transient? Only to some extent. Their effect or the force or the work that they do is minimal, but they act as a cofactor. Though if you see here, they have given coenzyme, but still the activity is in the form of cofactor only. The major role is played by apoenzyme only. Okay, next question here. This question was asked in the year 2015. Which one of the following statement is incorrect? Okay, what they are saying, which is incorrect. This question was asked in the year 2015 here. Okay, so we'll go one statement by statement. Substrate binds with enzymes at its active site. Okay, this is just an introduction that I'm giving you. Okay, now coming back to the options given here, look at option A. In competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. So this option is correct. Yes, in competitive inhibition, the inhibitor molecule is not chemically changed by the enzyme. Second option, the competitive inhibitor does not affect the rate of breakdown of enzyme substrate complex. That is also correct. Look at the third option here. The presence of the competitive inhibitor decreases the Km of the enzyme for the substrate. What do you mean by Km? Do you have any idea? What do you mean by Km enzymes? It's very important that you know. See, Km is a concentration of the substrate. Okay, what is Km? It denotes the concentration of substrate. That is why I told you in the beginning about substrate. Okay, before telling you the solution, I went directly, before telling you going into the options, I told you directly the answer. No problem, but I just gave you what is substrate. Likewise, Km, if you see, it is the concentration of the substrate which permits the enzyme to achieve half of Vmax. So usually they will represent in the form of graph. You must have seen. An enzyme with high Km has low affinity for its substrate and requires a greater concentration of the substrate to achieve Vmax. So here in the option C, they have said the presence of the competitive inhibitor decreases the Km of the enzyme. No. Only when the Km increases, we will achieve Vmax. Therefore, the correct answer is option C. Okay. It has to be maximum. They have said it decreases. No. Okay. Therefore, the correct answer is option C. Next question here. Select the option with correct, which is not correct with respect to the enzyme action. Again, we are talking about enzymes here. Enzymes are also called as biocatalysts. They are protein molecules. They help in aiding digestion and they play a major role in metabolic pathways. So these are some introduction about enzymes. Now we'll go one by one. Okay. First option, substrate binds with the enzyme at its active site. Yes, that is right. Okay, that is correct statement. Look at option B. Addition of lot of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. So here if you see, option B is only wrong. Okay, option B is only the right answer here. Inhibitions of succinic dehydrogenase by malonate is an example of competitive inhibition. What it is? Competitive inhibition. Okay, but here what they have given, addition of lot of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. Okay, but I told you inhibition of succinic dehydrogenase by malonate is an example of competitive inhibition. And if you see, they said it will not reverse. But actually, if you see, this process is reversible. Okay, so that is where they went wrong. Therefore, the correct answer is option B. This process which happens here is a reversible reaction. But in the option they have mentioned it is not reverse. Therefore, the correct answer to this question, which of the following is not correct? If you see, it is option B, which is not correct. Okay. Next question. This question asked in the year 2013, the essential chemical components of many coenzymes. Easy question for you to answer. See, when we talk about what do you mean by coenzyme, first you need to tell me that. There is something in addition with the coenzyme. That is, apart from enzyme, you also have something associated with it. A coenzyme is an organic non-protein compound that binds with an enzyme to catalyze a reaction. As I already said, co plus enzyme. Okay. with enzyme you also have something in association with it, so that it will help the reaction to get completed. You have the presence of this co. It can be a non-protein compound and it's organic only. Okay, coenzymes are often broadly called as cofactors, but they are chemically different. A coenzyme cannot function alone. As it is mentioned here, it is dependent. It will only go as a in action with any other ingredient. It cannot action alone. Therefore, coenzyme, if you see, will have majority of vitamins in it. Okay, it has vitamins in it. The essential chemical components of many enzymes are vitamins. Example, coenzymes, nicotinamide, adenine, dinucleotide. Then you have NADP. They all contain a vitamin called as niacin. So this is a very important vitamin that is found in many coenzymes. Okay, therefore the correct answer is vitamins. Okay, so I've given you a picture also to repeat that. Right, we have seen NADP here. Okay, next question here. This question was asked in the year 2013. Transition state structure of a substrate formed during an enzymatic reaction is dash. Again, this is an easy question for you. What do you mean by transition state of a substrate? No, tell me what is transition first. How can you describe transition? See, transition can be transition state or transition, whatever. The transition state of a chemical reaction is a particular configuration along the reaction coordinate. So what do we talk here? We talk about the reaction coordinate. Okay, we are talking about the reaction coordinate. Now, it is defined as a state corresponding to the highest potential energy along this reaction coordinate. It is often marked with a double dagger. It will be usually marked like this. It will be marked with a double dagger symbol to represent the transition state. So where we are talking about the transition state structure of a substrate formed during an enzymatic reaction is dash. The correct answer is option B. It is transient and unstable. Okay, unstable in the sense it is not stable, or it doesn't have the ability to be stable. And here if you see transient, they don't last for a long time. Okay, so that is the meaning of it. So I have explained to you what is transition state. It is often marked with a double dagger. It is a state of a chemical reaction in a particular configuration along the reaction coordinate. Therefore the correct answer is option B. Next question. This question was asked in the year 2013. Which of the following statements about the enzymes is wrong? So if you remember clearly, in the earlier part 2, we spoke a lot about RNA and DNA, right? Now if you look at part, I'm talking about the part 4. In part 3, we spoke about DNA and RNA a lot. Now in part 4, we are talking about enzymes in majority. If you clearly notice, you will understand and you will recognize it. Which one of the following statement about enzyme is wrong? So far I had been explaining what is enzyme and all that. Now, enzymes require optimum pH and temperature for maximum activity. Yes, they require that condition for it to attain its function. Enzymes are denatured at high temperatures. Yes, that is also true. Enzymes are highly specific. Option D is also right. Now look at option C. Enzymes are mostly protein, but some are lipids too. See, almost all enzymes will be proteins. Okay, majority, 99%, they are proteins. There are some nucleic acids that behave like enzymes. They are called as ribozyme. Okay, so even that 1%, if you see, it will be a mixture of protein in it. It will not be purely in the form of lipid and all. So enzymes are mostly protein, but some are lipids, which is something that cannot be agreed with. Therefore the correct answer is option C. Okay, next question. Look at this question here. This question was asked in the year 2011. The curve given below shows enzymatic activity with relation to 3 conditions: pH, temperature and substrate concentration. What do the two axes X and Y represent? So you just need to tell me what does it represent. Okay, so here they have given you temperature and substrate, and we have pH. All right. So what do you think will be X and Y? We're talking about X and Y, but we have 3 concentrations. If it is just 2, we could have mentioned it easily and go. But here if you see, the correct answer to this question is option B. Okay, temperature enzyme activity. Okay. See, in the diagram given below, the relation between temperature and enzyme activity is shown. Therefore, the correct answer is option B, temperature enzyme activity. pH, if you see, they already would have carried the experiment in some pH and they will plot the values. And here they are looking for the relation between the substrate and the temperature. pH is where they have already carried out the graph for the whatever process, or it can be any project, or it can be any material, any experiment that they have carried out. Now they have to check the relationship. We are not going to check the relationship with pH, but we have to check the relationship between temperature and substrate. Therefore, the correct answer is option B. Next question here, asked in the year 2008. A competitive inhibitor of succinic dehydrogenase. What do you mean by inhibitor? The name itself tells you, right? It inhibits something, right? So inhibit, the process to restrain or prevent, is the common meaning of inhibit. Now inhibitor, the one which will help in that inhibition activity. A competitive inhibitor of succinic dehydrogenase is, the correct answer is option A, malonate. So I've already almost given you. So what is this malonate? Okay, what do you know about malonate? Of course, it's a powerful inhibitor, but apart from that, what all you know about malonate? See, malonate can be described as, it's a powerful inhibitor. Apart from that, what you can say is, it is a compound that includes salts and esters. So what is it? What does it have? Salts plus esters. Okay. When we say salt and esters, what do they comprise? Where do this come from now? It comes from malonic acid. Okay, such as diethyl malonate, then you have dimethyl malonate and so on. Now, what is the use of this malonate? If you see, it is a 3-carbon dicarboxylic acid. It is also known as competitive inhibitor of succinate dehydrogenase. It occurs naturally in biological systems such as legumes and developing rat brains, which indicates that it may play an important role in symbiotic nitrogen metabolism and brain development. So malonate here, as they have mentioned, it's a powerful inhibitor of cellular respiration because it binds with the active site of the succinate dehydrogenase in the citric acid cycle, but it does not react. It will only inhibit, but it will not interfere with the reaction. Okay, it is thereby competes with succinate since it does not have a group, a succinate group. Okay, CH2, CH2, the dicarboxyl group is not present in it. Therefore, what happens? It will compete. Okay, so that is why it is called as a powerful inhibitor. It prevents or restrains the action. Therefore, the correct answer to this question is option A, malonate. Next question. This question was asked in the year 2006. Okay, let's read it here. An organic substance bound to an enzyme and essential for its activity is called as dash. This question asked in the year 2006. You have 4 options here. Right, an organic substance bound to an enzyme. I think all of you by now would have guessed the enzyme. It is coenzyme, because coenzyme is a cofactor cannot work or cannot act independently. We all know that. But a haloenzyme, apoenzyme, isoenzyme, they can do it independently. But here they are asking something that is bound to an enzyme and it is essential for its activity also. A cofactor cannot reside or cannot do its action independently. A coenzyme is an organic substance that enhances the action of the enzyme by binding it with a protein molecule, while haloenzyme is biologically or biochemically active compound formed by the combination of an enzyme with a coenzyme. So if you look at the rest of the other enzymes, excluding coenzyme out of it, all these are in association with certain enzymes, but the one which gets bound to the organic substance, one which gets to the an organic substance, a coenzyme, a cofactor can be a protein also. So one which binds to the enzyme is a coenzyme. Always remember that. Apoenzyme is the protein part of the enzyme. So here you can see here, apoenzyme is the protein part of the enzyme to which the coenzyme attaches to form an active enzyme. Isozyme is one of several forms of an enzyme that can catalyze the same reaction but differ from each other in many properties. So here they have clearly mentioned about an organic substance, right? Organic substance that will bind to an enzyme. So it is coenzyme. Always remember this. Okay. Next question, asked in the year 2005. Which of the following statement regarding the enzyme inhibition is correct? They're asking us which one is correct out of the 4 options given here. We'll go one option by option. Okay. Now here, if you see, competitive inhibition is seen when a substrate competes with an enzyme for binding to an inhibitor protein. This is right. We just now saw about the competitive inhibitor malonate. It will actually compete, so it's right. Next. Is it right? It will compete. But what else it does here? It competes, that is right. But apart from that, you also have something which is wrong here. With an enzyme for binding to an inhibitor protein, does it bind to an inhibitor protein there? No. Why does it compete there? Because of the absence of dicarboxyl group. So just because it has to bind to an inhibitor protein, it will not compete. The compete is correct, but the rest of the part is wrong. Therefore, the option A is not the right answer. Look at the next option. Competitive inhibition is seen when the substrate and the inhibitor compete for active site of the enzyme. So what are they competing for? They are competing for the active site on the enzyme. What does compete mean? They actually fight for it. Right. Therefore, the correct answer to this question is option B. In competitive inhibition, the inhibitor resembles the substrate in the structure and hence compete with the active site of the enzyme. Okay, option B is the right answer. Which statement of the 4 is correct if you see? Yes, why do they compete? They compete for the active site of the enzyme. They compete because they don't have the dicarboxyl group. When dicarboxyl group is there, they don't have to compete. Once they start compete, they have to compete for the active site of the enzyme. Okay, what is an active site of an enzyme now? How can you describe active site of an enzyme? Why do we call it active site? That is very important, right? Active site. See, when we say active site, that is where the real reaction begins. Right? We all know that. In biology, the active site is a region of an enzyme where the substrate molecule will bind to undergo a chemical reaction. Why? It will undergo to form a chemical reaction. Okay, the active site will consist of residues that form temporary bonds with the substrate and residues to catalyze the reaction of the substrate. So, what is that active site composed of? It is composed of the substrate which has to perform an action, which is about to perform an action. Okay, next question here. Again, 2005 question only this is. Let's read it here. The catalytic efficiency of 2 different enzymes can be compared with. I think by now you have guessed the answer. We already saw what is a Km enzyme, right? So the catalytic efficiency of 2 different enzymes can be explained or can be compared with the help of the Km value. Okay, it is called as Menton constant. It is defined as that substrate concentration at which under optimum conditions the rate of an enzyme is catalyzed reaction reaches half the maximum rate. And always remember that Km will be inversely proportional to the affinity of the enzyme. Okay, it will be inversely proportional to the affinity of the enzyme for its substrate. Apart from that, what else you can tell about the Km enzyme? If you see, I've already explained in the earlier questions as well. See, Km enzyme is a concentration of the substrate which permits the enzyme to achieve half of the Vmax. So our target is to achieve the maximum of Vmax. So what will help for that? If you see, it will help from the substrate value. Only when your Km increases, you will get the increase in Vmax also. Therefore, how do you compare? What do you compare? With the help of what? It is with the help of the Km value. Okay, next question here, which was asked in the year 2004. In which of the following enzymes is copper necessarily associated as an activator? They're asking us as an activator. Okay, see, we have 4 options here: carbonic anhydrase, tryptophanase, then you have lactic dehydrogenase, tyrosinase. Okay, tyrosinase. Now, out of the 4 options, the correct answer is tyrosinase. Tyrosinase is also called as polyphenol oxidase, and it's a copper containing oxidase which is widely distributed in plants, animals, and also it is present in man. What is its activity? If you see, its activity is to oxidize. It oxidizes tyrosine. Okay, it will oxidize tyrosine to pigment melanin in mammals and causes the cut surfaces of many fruits and vegetables to darken. So the browning effect, you must have seen the apples getting brown once they are exposed to air. So that is because of the oxidation that happens between the apple and the atmosphere. So that tyrosine. So the browning effect is because of this particular oxidation. Okay, so in which of the following enzymes is copper necessarily associated as an activator? It is tyrosinase, option D. Okay, so if you see many fruits and vegetables, they get darkened once they are exposed to the atmosphere. So that is because of this particular action. Okay, next question here. Feedback inhibition of an enzymatic reaction is caused by. So I'm going to show you a diagram so that it will be easy for you to understand. So here, if you see, we're talking about the feedback inhibition. Okay, what do you mean by feedback inhibition? Before we go into the answer, we should know what is feedback inhibition. Okay, see, feedback inhibition is a phenomenon where the output of the process is used as an input to control the behavior of the process itself. Understand it here. Feedback inhibition is a process or the phenomena where the output, okay, output of a process is used. Now the output itself is used as an input. Okay, it will be used as an input to control the behavior of the process itself. Although negative feedback is used in context of inhibition, negative feedback is also concerned about this particular process. So what is feedback inhibition in detail? If you have to tell, the output that comes itself is taken as an input to control the process. So the feedback inhibition of enzymatic reaction is caused by the end product. The output is nothing but they are denoting to the end product. Therefore the correct answer to this question is option A. Okay, fine. I think you have understood whatever is explained so far. So with this we complete part 4 of biomolecules. I hope all of you understood whatever was explained. In case you have any doubts, you can you can go through the video again so that you will get a clear information of it. Therefore we'll meet in the next part of the same chapter. Thank you.